Hello everyone. Again I am Girish Kumar Singh with you. Hope you are doing very well. Today I am preparing this slide to make you understand the instruments for studying cells. Dear students, cells are microscopic in structure and cannot be seen with a naked eye. So they are studied with the help of instruments, and the instruments are called microscopes. These provide enlarged images of cells and the structures they contain. So we can say microscope is the most common optical instrument used to observe cellular organization of living organisms. There are many different kinds of microscopes which can be broadly divided into two categories. The first is the light microscope and second is the electron microscope. In next slide, students, in this slide we will study about the light microscope. Dear all, it is commonly used in laboratory. It uses glass lenses and visible light that illuminate and passes through a specimen. Commonly used in laboratory. It uses glass lenses to magnify and visible light to illuminate to shine, and passes through a specimen. Two kinds of light microscope used frequently in the laboratory, and they are simple microscope and compound microscope. In this slide, the figure which you are seeing at the slide is of simple microscope. Simple microscope are just magnifying glasses. In fact, human eye works as an optical instrument and the eye lens acts as a simple lens. The resolving power of a healthy young human eye is approximately 0.1 millimeter at 25 centimeter viewing distance. That's why whenever you have to study to see your books, maintain the distance at least 25 centimeter from the eyes to books. Any object smaller than this cannot be viewed by naked eye. A microscope is therefore used for enlargement and magnifying the smaller objects so that it becomes visible. Dear students, this slide is showing you the diagram of a compound microscope. It is one in which the magnification takes place in two stages. It consists of two lens units. The first lens unit, called objective, produces a primary magnified image, and the second lens unit, that is called the eyepiece or ocular, magnifies the first image. Such microscopes employing two lenses units in working position at one time are called monoocular mono-objective microscope. Mono means one ocular, that is eyepiece, and mono-objective, one is objective lens, and both are working together at same time. So it is called monoocular mono-objective microscope. The simple student's microscope, generally used in biological laboratory to observe tiny cells, is called compound microscope. Such a microscope uses light, generally sunlight, to illuminate the object or specimen under study, and hence it is also called light microscope. It has the magnification capacity of 100 times to 1500 times. Means the image of the object can be seen 100 times to 1500 times more bigger than the actual object. To magnify the object, power of objective. Objective has 3 magnification power. One is of 10 times magnification, second is the 40 times of magnification, and third one is the 100 times of magnification, and all are attached to a nose piece that are movable. You can adjust any one of them, and eyepiece that has 2 replaceable pieces that have the 10 times and 15 times of magnification. Suppose you are using the magnification capacity of objective 10 times and also the 10 times magnifying capacity of eyepiece, then you will see the image 100 times bigger. Similarly, you can use them one by one. The light or compound microscope is a strong heavy metal instrument. We are talking about the structure. It comprises a U-shaped base having 2 vertical pillars. A curved arm is movably joined to the pillars to hold the microscope. The arm can be bent over the pillars at inclination joint to shoot the viewer, whatever is the comfortable for viewer. The upper part of the arm holds the movable body tube. The other part of the microscope are reflector, condenser, lens, stage, objective lens, ocular lens that is also called eyepiece, and adjustment screws. There are 2 types of adjustment screws, coarse and fine. A glass side containing the object or specimen under study is kept on a stage under an objective lens. Light is then passed through the object with the help of a mirror and the condenser from below the stage. A magnified sharp image of object can thus be seen through the ocular lens by focusing the coarse adjustment. Keep on seeing on diagram. And fine adjustment knobs properly. We can increase or decrease the magnification of the image by changing the objective lens of high or low power. Dear students, you are seeing the diagram of electron microscope. It is a very large instrument that helps in observing subcellular structures which cannot be seen with light microscope. It uses very high voltage electricity for its working. For its working, internal vacuum is essential. It uses electromagnets instead of glass lenses and beam of electrons instead of light. Ultra thin and dry section of material is impregnated with some metal to enhance contrast, and the image is obtained on a photographic plate or fluorescent screen. Magnification capacity of this lens is 1 lakh to 5 lakh times, and this microscope has been discovered by Knoll and Ruska in 1940. Dear one, as you know, this was Girish Kumar Singh speaking to you, and hope you will able to understand the instruments for studying cells. If you have any problem, feel free to ask. Till then, stay home, remain safe, and prepare for a new battle. Thank you very much. Have a nice day.